Hello friends, this is Dr. Ashish Sharma and I welcome you again in my YouTube channel Concepts Connect. In this video, I will talk about different titrimetric methods of chemical analysis. This video is especially prepared for UG students where they will get the comparative summary of all the major titrimetric analysis including neutralization titrations, Redox-Titrations, Complexometric Titrations, Precipitation Titrations, and Gravimetric Titrations. This is the obvious question that have to ask ourselves that why we should study titrimetric analysis. What types of industries are actually there we can find jobs in, especially after being skilled in titrimetric analysis. Yes, there are different types of industries where titrations are being used as the main method of chemical analysis. Of course, pharmaceutical industries are one of the types of such industries where active pharmaceutical ingredients or API or drugs are being analyzed quantitatively by different titrimetric analysis. Here, I use two new terms. One is active pharmaceutical ingredients, which means a drug, and another term is quantitative analysis, which means to find out the quantity of a drug. For example, finding out quantity of acetyl salicylic acid in aspirin or vitamin C in multivitamins tablets. The other types of industries where titrimetric analysis are used are food industries, cosmetic industry, wine industry, Carl Fisher titration, biodiesel manufacturing, medical diagnosis to name a few. Titrations are also used in acid rain analysis and wastewater management. Examples of uses of titrations associated with particular type of industries are mentioned on the slides for understanding. Alright, now I believe that you understand the importance of titrations. Now the first question is, what is titration? So, titration is a technique of chemical analysis in which a solution of non-concentration is used to find out the concentration of a non-solution by using an indicator. Generally, in titrations, we add a solution of non-concentration from burette to the solution of a non-concentration and allow them to react completely. Once the titration is completed, we stop adding solution of non-concentration from burette. The point when reaction gets completed is called the end point. Now the question is, how would you know that we have achieved the end point? The answer is indicator. The indicators change their color to indicate the end point where we can stop adding titrant immediately from burette. All the titrations always have three components. One is solution of non-concentration, which is also known as titrant and generally taken in burette. Another component is the solution of a non-concentration, which is called analyte or titrant. And the third component is indicator, which tells us about the end point. Titrations is also known as volumetric analysis because we find out volume of titrant or standard solution which react with analyte. Here, I would like to let you know about primary and secondary standard solutions also. Substance of non-high purity, 99.9% pure, are called primary standard, and solution of primary standards are non-primary standard solutions. And these solutions are generally used as titrant, while secondary standard solutions must be standardized with respect to primary standards before using as titrant. If you want me to prepare a separate video on standardization of secondary standards, please mention in the comment box. This slide shows a glimpse of major type of titrimetric analysis based on the reaction involved between titrants and analytes. First type of titration is acid-base titration, which involves acid-base reactions or neutralization reactions. Second type of titration is redox titration, which involves oxidation reduction reactions between titrants and analytes. Third type of titrimetric method is complexometric titration, which involves complex formation between ethylene diamine tetraacetic acid, which is EDTA, and metal ions through coordination bonds or electron pair donation. Precipitation titration are the fourth type of volumetric titrations, which involve precipitation formation between titrants and analytes. And fifth type of titrations are gravimetric titrations, in which precipitation formed between analytes and titrants is filtered, dried, and weighed. In the coming part of this video, I will letting you know about the details of these 5 titrimetric methods. In this slide, I would talk about acid-base titrations. In these titrations, either an acid neutralizes a base or a base neutralizes an acid to form salt and water. Hence, these titrations are also known as neutralization titrations. Acids are always titrated with bases and bases are always titrated with acids. Here, I am presenting two examples of acid-base titrations. In example 1, NaOH is being titrated or standardized against either potassium hydrogen phthalate or oxalic acid. Here, standardization of NaOH means finding out normality or molarity of the NaOH solution. As NaOH is an aqueous base, it has to be titrated with aqueous acids only. Hence, we are using potassium hydrogen phthalate or oxalic acid as acids and as primary standards. Why primary standards? Because purity of both potassium hydrogen phthalate and oxalic acid is higher than 99.9%. In this case, NaOH is an analyte, as we are finding out its strength or concentration, and is taken in conical flask. Potassium hydrogen phthalate or oxalic acid act as a titrant, as their concentrations are exactly known to us. Apart from titrant and analyte, we need an indicator which will let us know about endpoint. Phenolphthalein is used as indicator in this case, and colorless to pink color of phenolphthalein indicates the endpoint. Another example of neutralization titration is standardization of HCl or H2SO4 with solution of sodium carbonate. In this titration, solution of HCl or H2SO4 taken in conical flask acts as analyte or titrant, because we are finding out unknown concentration of HCl or H2SO4 solutions. Solution of sodium carbonate is used as primary standard and titrant and is taken in burette. Methyl red is used as indicator in this case. In this slide, I would explain redox titrations in brief. In redox titration, oxidation-reduction reactions occurs between analytes and titrants. We will understand redox titrations with the help of two examples. First one is the standardization of potassium permanganate KMnO4. In this example of redox titration, KMnO4 acts as an analyte, self-indicator and oxidizing agent. Here it acts as an analyte or secondary standard only because it is difficult to obtain the pure state of KMnO4 as it is not free from MnO2. Oxalic acid acts as an titrant or primary standard and a reducing agent in this case. This titration occurs in acidic medium, that is why we are using H2SO4. Potassium permanganate oxidizes oxalic acid in presence of acid and it gets converted into manganese sulfate. In this reaction, MnO4 ions of potassium permanganate change to Mn+2 ions. Here, what happens? Potassium permanganate is of purple color due to MnO4. When it is added to acidic solution of oxalic acid, it is a reducing agent. Permanganate solution decolorized to a colorless solution because MnO4 ions of potassium permanganate changes to Mn+2 ion, which are colorless. Once all the oxalic acid gets consumed or reacted with potassium permanganate, then the next drop of potassium permanganate does not find any oxalic acid to react with it and remains intact, which gives the purple pinkish color to solution. In this way, potassium permanganate acts as self-indicator. In this example, we are standardizing potassium permanganate, hence it is being taken as analyte. But standardized potassium permanganate can be used as titrant, as we can see in the next example of redox titration mentioned here. Standardized potassium permanganate is used as titrant to perform assay of ferrous sulfate. In this titration also, potassium permanganate acts as oxidizing agent and self-indicator, while ferrous sulfate acts as reducing agent and analyte. In this titration also, endpoint is detected due to self-indicating property of potassium permanganate. This slide talks about complexometric titration. This term complexometric itself suggests that there is some complex formation occurs during titration. What sort of complex forms during these titrations? Answer is complex formation occurs between ethylene diamine tetraacetic acid and the metal ions. Conceptually, these complexes are ligand metal ion chelates. Here, ethylene diamine tetraacetic acid acts as a ligand. More precisely, EDTA is a hexadentate ligand, which means it can form 6 bonds with a metal ion by donating 6 pairs of non-bonding electrons. Form the red circled group you can see in the slide and form the metal ion complex. In these type of titrations, ethylene diamine tetraacetic acid acts as titrant, chelating agent or complex forming agent and as a primary standard and taken into a burette, while metal ions acts as analyte, for example, calcium ions and magnesium ions. Different indicators are used to detect the endpoint in complexometric titrations are calcine, curcumin, eriochrome black tea, fast sulfone black, hematoxylin, etc. The classic use of complexometric titration is to determine the hardness of water, because majority of the ions which are responsible for hardness of water is calcium ions, and concentration of calcium ions can easily be determined by complexometric titrations. Next type of titration I am going to discuss here is precipitation titrations. Name precipitation itself indicates that precipitation reactions are involved into these titrations. In this type of titrations, titrant reacts with the analyte and forms an insoluble substance or precipitate. Titration is continued till all the amount of analyte is consumed. The slight axis of titrant reacts with the indicator and gives signal to terminate the titration process. The precipitation titrations I am highlighting here are Argentometric titrations. Argentometric titrations are those precipitation titrations in which silver ions or argentum ions are involved. Argentometric titrations can be classified into three types: Morse method, Volhard method, and Fezan's method. This slide is showing the first type of Argentometric titration, which is performed by Morse method. In this method, silver nitrate is used as titrant and chloride ions are used as analyte. Potassium permanganate is used as an indicator. At the end point, when all the chloride ions are consumed by silver ions, the slight axis of silver nitrate results into reddish brown colored precipitate of silver chromate, which is formed by reaction of silver and chromate ions. This is a direct method of quantitative analysis of silver ions by argentometric titration. This slide shows the second type of argentometric titration, which is Vollard's method. This is a two-step method. In the first step, halide ion solution or any other anionic solution acts as first analyte, which is titrated with the measured axis of AgNO3, which acts as a titrant. The reaction involved into this step is shown in equation 1. This measured axis of silver ion is used as analyte in the second step and titrated with the standard solution of potassium thiocyanate. At the end point, when all the silver ions are consumed by thiocyanate ions, the next drop of potassium thiocyanate reacts with ferric ion indicator and develop a red color solution, which is due to the formation of the ferrous thiocyanate compound. Kindly see the equation number 2 and 3. The concentration of chloride ions is determined by subtracting the titration findings of the moles of silver ions that reacted with the thiocyanate from the total moles of silver nitrate added to the solution. This is the indirect method of titration. This slide shows the third type of argentometric titration, which is performed by Fezan's method. This is also known as indicator adsorption method. In this method, chloride ions present in excess are adsorbed on precipitate of silver chloride surface. The surface of AgCl precipitate becomes negatively charged, and then the positively charged silver ions of titrant AgNO3 get adsorbed onto it. Thus the surface of precipitate becomes positively charged now. Dichlorofluorescein is used as an indicator in this titration. This is a negatively charged compound and makes ion pair with positively charged silver ions to give green color to precipitate. The end point is determined by pink color of complex of AgCl and indicator as shown in red square. This is also a direct method of precipitation titration. The next and last type of titration included in this video is gravimetric titration. In this method of analytical chemistry, the quantity of an analyte based on the mass of a solid is determined. In this type of analysis, a precipitating agent is added to a solution of analyte to precipitate the analyte of interest. We have to be sure that none of the analyte's molecule remains unprecipitated. For assuring this, a slight excess of precipitating agent is added to analyte solution. Thus formed precipitate is filtered, washed, dried and weighed to find the quantity of the analyte in a solution. Millions of thanks for watching this video. If you feel this video is useful, kindly share and like the video. Subscribe Concepts Connect YouTube channel for being updated with the upcoming videos. Thank you so much once again.